Hello everyone, Professor Baida here. Let's examine how cells express their gene. So we have chapter 17, all about gene expression from gene to protein. Inherited traits like albinism are determined by genes, which have information content in the form of specific sequences of nucleotides along stretches of DNA, the genetic material. The DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins and of RNA molecules involved in protein synthesis. In the case of coat and skin color, the gene containing the information of synthesize an enzyme that makes pigment allows normal coloring. And a faulty version of that gene leads to a lack of color or the albino phenotype. Proteins are the link between genotype and phenotype. Gene expression is the process by which DNA directs the synthesis of protein, or in some cases, just RNAs. The expression of genes that code for proteins include two stages, transcription and translation. Genes provide the instructions for making specific proteins, but a gene doesn't build a protein directly. The bridge between DNA and protein synthesis is the nucleic acid RNA. This type of RNA molecule is called messenger RNA. Transcription is the synthesis or production of the RNA using information in DNA. The two nucleic acids are written in different forms of the same language, and the information is simply transcript or written from DNA to RNA. This type of RNA molecule is called messenger RNA because it carries a generic message from the DNA to the protein synthesizing machinery of the cell. Translation is the synthesis of a polypeptide using the information in the mRNA. During this stage, there is a change in language. The cell must translate the nucleotide sequence of an mRNA molecule into the amino acid sequence of a polypeptide. The site of translation are ribosomes, molecular complexes that facilitate the orderly linkage of amino acid into polypeptide chains. Transcription and translation occur in all organisms because most studies have involved bacteria and eukaryotic cells. The basic mechanisms or the basic mechanics of transcription and translation are similar for bacteria and eukaryotes, but there is an important difference in the flow of genetic information within the cells. Bacteria don't have nuclei. Therefore, nuclear membrane don't separate bacterial DNA and mRNA from ribosomes and other protein synthesizing equipment. The lack of compartmentalization allows translation of an mRNA to begin while its transcription is still in progress. So as you see here, in bacterial cell, which lacks a nucleus, mRNA produced by transcription is immediately translated without additional processing. By contrast, eukaryotic cells have nuclei. The presence of nuclear envelope separates transcription from translation in space and time. Transcription occurs in the nucleus, but the mRNA must be transcribed or transported to the cytoplasm for translation. In eukaryotes, before RNA transcripts from protein coding genes can leave the nucleus, they are modified in various ways to produce the final functional mRNA. The transcription of protein coding eukaryotic gene results in pre-mRNA, and further RNA processing yield the finished mRNA. So in eukaryotic cell, the nucleus provides a separate compartment for transcription. The original RNA transcript called pre-mRNA is processed in various ways before leaving the nucleus as mRNA. The initial RNA transcript from any gene, including those specifying RNA that is not translated into proteins such as tRNA and RNA, rRNA, is more generally called a primary transcript. To summarize, genes program protein synthesis via genetic messages in the form of messenger RNA. Put another way, cells are governed by a molecular chain of command with a directional flow of genetic information, DNA, RNA, protein. So DNA transcript to RNA and then translated to protein. This idea that the flow of information went only one way was named the central dogma by Francis Crick in 1956. When biologists began to suspect that the instructions for protein synthesis were encoded in DNA, they recognized a problem. There are only 4 nucleotide bases to specify 20 amino acids. So how many nucleotides then would turns out to correspond to an amino acid? Triplets of nucleotide bases are the smallest units of uniform length that can code for all the amino acids. Uh, if each arrangement of 3 consecutive nucleotide bases specifies an amino acid, there can be 46 possible code words, more than enough to specify all the amino acids. Experiments have verified that the flow of information from gene to protein is based on a triplet code. The genetic instructions for a polypeptide chain are written in the DNA as a series of non-overlapping 3 nucleotide words. The series of words in a gene is transcribed into a complementary series of non-overlapping 3 nucleotide words in mRNA, which is then translated into a chain of amino acids. During transcription, the gene determines the sequence of nucleotide bases along the length of RNA molecule that is being synthesized. For each gene, only one of the 2 DNA strands is transcribed. This strand is called the template strand because it provides the pattern to or template for the sequence of nucleotides in the RNA transcript. For any given gene, the same strand is used as the template every time that gene is transcribed. The mRNA nucleotide triplets are called codons, and they are customarily written in the 5' to 3' direction. The term codons is also used for the DNA nucleotide triplets along the non-template strand. These codons are complementary to the template strand and thus identical in sequence to the mRNA except for thiamines instead for uracils. For this reason, the non-template DNA strand is often called the coding strand. By convention, the sequence of coding strand is used when a gene sequence is reported. During translation, the sequence of codons along on an mRNA molecule is decoded or translated into a sequence of amino acids making up a polypeptide chain. The codons are read by the translation machinery in the 5' to 3' direction along the mRNA. Molecular biologists cracked the genetic code of life in the early 1960s when a series of elegant experiments disclosed the amino acid translation of each of the mRNA codons. Although more elaborate techniques were required to decode mixed triplets such as AUA and CGA, all 64 codons were deciphered by the mid-1960s. 61 of the 64 triplet codes for amino acids. The other three codons act as stop signals or termination codons, marking the end of translation. The codon AUG has a dual function. It codes for the amino acid methionine and also functions as a start signal or initiation codon. Generic messages usually begin with the mRNA codon AUG, which signals the protein synthesizing machinery to begin translating the mRNA at that location. The three nucleotide bases of an mRNA codon are designated here as the first, second, and third bases, reading in the 5' to 3' direction along the mRNA. The codon AUG not only stands for the amino acid methionine, but also functions as a start signal for ribosomes to begin translating the mRNA at that point. 3 of the 64 codons functions as a stop signals, marking where ribosomes end translation. The genetic code is nearly universal, shared by organisms from the simplest bacteria to the most complex plants and animals. In laboratory experiments, genes can be transcribed and translated after being transplanted from one species to another, sometimes with quite striking results as shown in this figure. A bacteria can be programmed by the insertion of human genes to synthesize certain human proteins for medical use, such as insulin. A tobacco plant expressing a firefly gene, the yellow glow is produced by a chemical reaction catalyzed by the protein product of the firefly gene. A mosquito larva expressing a jellyfish gene, the green codes for green fluorescent protein and is inserted into organisms as a reporter gene, so researchers can tell if a gene of interest is being expressed. Messenger RNA, the carrier of information from DNA to the cell's protein synthesizing machinery, is transcribed from the template strand of a gene. An enzyme called RNA polymerase detach the two strands of DNA apart and joins together RNA nucleotides complementary to the DNA template strand, thus elongating the RNA polynucleotide. Like the DNA polymerases that function in DNA replication, RNA polymerase can assemble a polynucleotide only in its 5' to 3' direction, adding onto its 3' end. Unlike DNA polymerases, however, RNA polymerases are able to start a chain from scratch. They don't need to add the first nucleotide onto pre-existing primer. So the three stages of transcription are initiation, elongation, and termination of the RNA chain. The stages of transcription, initiation, elongation, and termination are shown in this figure. This general depiction of transcription applies to both bacteria and eukaryotes. In bacterium, the RNA transcript is immediately usable as mRNA. In eukaryote, the RNA transcript must first undergo processing. Initiation, after RNA polymerase binds to the promoter, the polymerase unwinds the DNA strands and initiate RNA synthesis at the start point on the template strand. In elongation, the polymerase moves downstream, unwinding the DNA and elongating the RNA transcript from 5' to 3' direction. After transcription has occurred, the DNA strands reform a double helix. In termination, eventually the RNA transcript is released and polymerase detach from the DNA. A specific sequence of nucleotides along the DNA mark where transcription of a gene begins and ends. The DNA sequence where RNA polymerase attach and initiates transcription is known as the promoter. So the promoter sequence of a gene includes within the transcription start point, the nucleotide where RNA polymerase actually begins synthesizing the mRNA and typically extends several dozen or so nucleotide pairs upstream from the start point. In eukaryotes, a collection of protein called transcription factors help guide the binding of RNA polymerase and the initiation of transcription. Only after a transcription factor are attached to the promoter does RNA polymerase bind to it. The whole complex of transcription factors and DNA polymerase II bind to the promoter is called transcription initiation complex. The role of transcription factors and a crucial promoter DNA sequence called the Tata box, informing the initiation complex at a eukaryotic promoter. This figure illustrated the initiation of transcription at eukaryotic promoter. In eukaryotic cells, proteins called transcription factors mediate the initiation of transcription by RNA polymerase II. A eukaryotic promoter commonly includes a Tata box, a nucleotide sequence containing a Tata about 25 nucleotides upstream from the transcriptional start point. Several transcription factors, one recognizing the Tata box, must bind to the DNA before RNA polymerase II can bind to the correct position and orientation. Additional transcription factor, purple color, bind to the DNA along with RNA polymerase II, forming the transcription initiation complex. RNA polymerase II then unwind the DNA double helix and RNA synthesis begins at the start point on the template strand. As RNA polymerase moves along the DNA, it untwists the double helix, exposing about 10 to 20 DNA nucleotides at a time for pairing with RNA nucleotides. The enzyme adds nucleotides to the 3' end of the growing RNA molecule as it moves along the double helix. As transcription proceeds forward, the newly synthesized RNA molecule behind the RNA polymerase peels away from its DNA template and the DNA double helix reforms. Transcription progress at a rate of about 40 nucleotides per second in eukaryotes. A single gene can be transcribed simultaneously by several molecules of RNA polymerase following each other like tracks in a convoy. In this image, RNA polymerase moves along the DNA template strand, joining complementary RNA nucleotides to the 3' end of the growing RNA transcript behind the polymerase. The new RNA peels away from the template strands, which reforms a double helix with the non-template strand. Bacteria and eukaryotes differ in the way they terminate transcription. In bacteria, transcription proceeds through a terminator sequence in the DNA. The transcript terminator, an RNA sequence, functions as the termination signal, causing the polymerase to detach from the DNA and release the transcript, which requires no further modification before translation. In eukaryotes, RNA polymerase II transcribes a sequence on the DNA called the polyadenylation signal sequence, which specifies a polyadenylation signal It's A in the pre-mRNA. This is called a signal because once this stretch of 6 RNA nucleotides appear, it is immediately bound by certain proteins in the nucleus. Then at a point about 10 to 35 nucleotides downstream from the AAA, these proteins cut the RNA transcript free from the polymerase, releasing the pre-mRNA. The pre-mRNA then undergoes processing, the topic of the next section. We will talk about it. And after that, cleavage marks the end of the mRNA. The RNA polymerase II continues to unscribe enzymes to degrade the RNA made after cleavage, starting at its newly exposed 5' end. The polymerase continues transcribing, pursued by the enzymes until they catch up to the polymerase and it dissociate from the DNA. Eukaryotic cells modify RNA after transcription. Enzymes in the eukaryotic nucleus modify pre-mRNA in specific ways before the genetic message is dispatched to the cytoplasm. During this RNA processing, both ends of the primary transcript are altered. Also, in most cases, certain interior sections of the RNA molecule are cut out and the remaining parts spliced together. This modification produces an mRNA molecule ready for translation. Alteration of mRNA ends include each end of a pre-mRNA molecule is modified in a particular way. Enzymes modify the two ends of the eukaryotic pre-mRNA molecule. The modified ends may promote the export of mRNA from nucleus, and they help protect the mRNA from degradation when the mRNA reaches the cytoplasm. The modified ends in conjunction with the certain cytoplasmic proteins help with ribosome attachment. The 5' cap and poly A tail are not translated into a protein, nor are the regions called the 5' untranslated region and 3' untranslated region. So the 5' end, which is synthesized first, receive a 5 cap modified form of guanine nucleotide added onto the 5' end after transcription of the first 2 to 40 nucleotides have been transcribed. At the 3' end, an enzyme then adds 50 to 250 adenine or more from adenine nucleotides forming a poly A tail. So on 5' end we have 5' cap, and in the 3' end we have poly A tail. So the 5' cap and poly A tail share several important functions. First, they seem to facilitate the export of the mature mRNA from the nucleus. Second, they help protect the mRNA from degradation by hydrolytic enzymes. Third, they help ribosomes attach to the 5' end of the mRNA once it reaches the cytoplasm. A remarkable stage of RNA processing in the eukaryotic nucleus is called RNA splicing, in which large proteins of the RNA primary transcript molecule are removed and the remaining protein are reconnected. The sequence of DNA nucleotides that codes for eukaryotic polypeptide is usually not continuous, it is split into segments. The non-coding segment of nucleic acid that lie between coding regions are called intervening sequences or introns. The other region are called exons. Because they are eventually expressed, usually by being translated into amino acid sequences. In making a primary transcript from a gene, RNA polymerase II transcribes both introns and exons from the DNA, but the mRNA molecule that enters the cytoplasm is in a bridge version. In the process of RNA splicing, the introns are cut out from the molecule and the exons joined together, forming an mRNA molecule with a continuous coding sequence. How is pre-mRNA splicing carried out? The removal of introns is accomplished by a large complex made of proteins and small RNAs called a spliceosome. Spliceosome, this complex binds to several short nucleotide sequences along an intron, including key sequences at each end. The intron is then released and rapidly degraded, and the spliceosome join together the two exons that flanked the introns. Ribozymes, the idea of catalytic role for the RNAs in the spliceosome arose from the discovery of ribozymes, RNA molecules that function as enzymes. Some introns contain sequences that regulate gene expression, and many affect gene products. One important consequence of the presence of introns in genes is that a single gene can encode more than one kind of polypeptide. Many genes are known to give rise to two or more different polypeptides, depending on which segments are treated as exons during RNA processing. This is called alternative RNA splicing. Proteins often have a modular architecture consisting of discrete structural and functional regions called domains. One domain of an enzyme, for example, might include the active site, while another might allow the enzyme to bind to a cellular membrane. In quite a few cases, different exon code for the different domains of a protein. Genetic information flows from mRNA to protein through the process of translation. As a molecule of mRNA is moved through a ribosome, codons are translated into amino acids one by one. The translator or interpreter are tRNA molecules, each type with a specific anticodon at one end and corresponding amino acid at the other end. A tRNA adds its amino acid cargo to a growing polypeptide chain with the anticodon. Hydrogen bonds to the complementary codon on the mRNA. In the process of translation, a cell reads a generic message and builds a polypeptide accordingly. The message is a series of codon along on mRNA molecule, and the translator is called transfer RNA or tRNA. The function of tRNA is to transfer an amino acid from the cytoplasmic pool of amino acids to a growing polypeptide in a ribosome. A cell keeps its cytoplasm stocked with all 20 amino acids, either by synthesizing them from other compounds or by taking them up from the surrounding solution. The ribosome, a structure made of proteins and RNAs, adds each amino acid brought to it by tRNA to the growing end of the polypeptide chain. tRNA molecule consists of a single RNA strand that is only about 80 nucleotides long. The presence of complementary stretches of nucleotide bases that can hydrogen bond to each other allows this single strand to fold back on itself and form a molecule with a particular 3D structure or three-dimensional structure. Flattened into one plane to clarify this base pairing, a tRNA molecule looks like a clover leaf. The tRNA actually twists and folds into a compact 3D structure that is roughly L-shaped with the 5' end and 3' ends of the linear tRNA both located. We have 3' end and 5' ends located here, one end of the structure. The protruding 3' act as the attachment site for an amino acid. The loop extending from the other end of the L shape includes the anticodon, the particular nucleotide triplets that base pairs to a specific mRNA codon. Thus, the structure of tRNA molecule fits its function. Although the ribosomes of bacteria and eukaryotes are very similar in structure and function, eukaryotic ribosomes are slightly larger and differ somewhat from bacterial ribosomes in their molecular composition. The differences are medically significant. Certain antibiotic drugs can inactivate bacterial ribosomes without affecting the ability of eukaryotic ribosome to make proteins. Ribosomes facilitate the specific coupling of tRNA anticodon with the mRNA codon during protein synthesis. A ribosome consists of a large subunit and small subunit, each made up of proteins and one or more ribosomal RNA or rRNA. A schematic model showing binding sites. Ribosomes has an mRNA binding site and 3 tRNA binding sites, known as the A, P, E sites. And this schematic model with mRNA and tRNA, a tRNA fits into the A site when its anticodon base pairs with an mRNA codon. The P site holds the tRNA attached to the growing polypeptide. The A site holds the tRNA carrying the next amino acid to be added to the polypeptide chain. Discharged tRNA leave from the E site. The polypeptide grows at its carboxyl end. We can divide translation, the synthesis of polypeptide, into 3 stages: initiation, elongation, and termination. All 3 require protein factors that aid the translation process. Some steps of initiation and elongation also require energy provided by hydrolysis of guanosine triphosphate GTP. A small ribosomal subunit binds to an mRNA in a bacterial cell. The mRNA binding site on this subunit recognizes a specific nucleotide sequence on the mRNA just upstream from the start codon. An initiator tRNA with the anticodon UAG base pairs with the start codon AUG. This tRNA carries the amino acid methionine. The arrival of a large ribosomal subunit completes the initiation complex. Proteins called initiation factors, it's not shown here, are required to bring all the translation components together. Hydrolysis of guanosine triphosphate provides the energy for the assembling. The initiator tRNA is in the P site. The A site is available to the tRNA bearing the next amino acid. In the elongation stage of translation, amino acids are added one by one to the previous amino acid at the C-terminus of the growing chain. Each addition involves several proteins called elongation factors and occurs in a three-step cycle: uh, codon recognition, peptide bond formation, and translocation. Energy expenditure occurs in the first and third steps. The mRNA is moved through the ribosome in one direction only, 5' end first. This is equivalent to the ribosome moving 5' to 3' on the mRNA. In this schematic model, codon recognition, the anticodon of an incoming amino acid tRNA base pairs with the complementary mRNA codon in the A site. Hydrolysis of guanosine triphosphate increases the accuracy and efficiency of this step. Although not shown, many different amino acid tRNA are present, but only the one of the appropriate anticodon will bind and allow the cycle to progress. Here in the second step, peptide bond formation, an rRNA molecule of the large ribosomal subunit catalyzes the formation of peptide bond between the carboxyl end of the growing polypeptide in the P site and the amino group of the new amino acid in the A site. As shown in the next diagram, this step removes the polypeptide from the tRNA in the P site and attaches it to the amino acid on the tRNA in the A site. In translocation, the ribosome translocates the tRNA in the A site to the P site. At the same time, the empty tRNA in the P site is moved to the E site, where it is released. The mRNA moves along with its bond tRNAs, bringing the next codon to be translated into the A site. The final stage of translation is termination. Elongation continues until a stop codon in the mRNA reaches the A site of the ribosome. A release factor, a protein shaped like an amino acid tRNA, binds directly to the stop codon in the A site. This is the stop codon. When a ribosome reaches a stop codon on mRNA, the A site of the ribosome accepts a release factor, a protein shaped like tRNA instead of an amino acid tRNA. The release factor promotes hydrolysis of the bond between the tRNA in the P site and the last amino acid of the polypeptide, thus freeing the polypeptide from the ribosome. The two ribosomal subunits and the other components of the assembly dissociate with the hydrolysis of 2-guanosine triphosphate molecule. During its synthesis, a polypeptide chain begins to coil and fold spontaneously as a consequence of its amino acid sequence primary structure, forming a protein with a specific shape, a three-dimensional molecule with secondary and tertiary structure. Thus, a gene determines primary structure, which in turn determines shape. Additional steps, post-translational modifications, may be required before the protein can begin doing its particular job in the cell. Certain amino acids may be chemically modified by the attachment of sugars, lipids, phosphate groups, or other addition. An electron micrographs of eukaryotic cells active in protein synthesis, two populations of ribosomes are evident: free and bound. Free ribosomes are suspended in the cytosol or cytoplasm and mostly synthesize proteins that stay in the cytosol and function there. In contrast, bound ribosomes are attached to the cytosolic side of the endoplasmic reticulum or to the nuclear envelope. Bound ribosomes make proteins of the endomembrane system as well as proteins secreted from the cell, such as insulin. It is important to note that the ribosomes themselves are identical and can alternate between being free ribosomes one time they are used and being bound ribosomes the next. Targeting polypeptide to specific locations, polypeptide synthesis begin on a free ribosome in the cytosol, and a signal recognition particle, SRB, signal recognition particle, binds to the signal peptide if present, halting synthesis momentarily. The signal recognition particle binds to the receptor protein in endoplasmic reticular membrane, part of a protein complex that forms a pore. The signal recognition particle leaves and polypeptide synthesis resumes with simultaneous translocation across the membrane. Here, the signal peptide is cleaved by an enzyme in the receptor protein complex, and the rest of a completed polypeptide leaves the ribosome and fold into a final conformation. In both bacteria and eukaryotes, multiple ribosomes translate an mRNA at the same time. That is, a single mRNA is used to make many copies of polypeptide simultaneously. Once a ribosome is far enough past the start codon, a second ribosome can attach to the mRNA, eventually resulting in a number of ribosomes trailing along the mRNA. Such string of ribosomes, called polyribosomes or polysomes, can be seen with an electron microscope. They can be either free or bound. They enable a cell to rapidly make many copies of a polypeptide. Another way both bacteria and eukaryotes increase the number of copies of polypeptide is by transcribing multiple mRNA from the same gene. However, the coordination of two processes, transcription and translation, differs in two groups. The most important difference between bacteria and eukaryotes arises from bacterial cells' lack of compartmental organization, like a one-room workshop. This image shows an mRNA molecule is generally translated simultaneously by several ribosomes in cluster called polysomes or polyribosomes. A bacterial cell ensures a streamlined operation by coupling the two processes with no nuclear envelope. It can simultaneously transcribe and translate the same gene, and the newly made protein can quickly diffuse to its site of function. This image in bacterial cell, the translation of mRNA can begin as soon as the leading 5' end of the mRNA molecule peels away from the DNA template, the DNA being transcribed by RNA polymerase molecules attached to each RNA polymerase molecule is the growing strand of mRNA and which is already being translated by ribosomes. In contrast, the eukaryotic cell's nuclear envelope segregates transcription from translation and provides a compartment for extensive RNA processing. This diagram shows the path from one gene to one polypeptide. Each gene in the DNA can be transcribed repeatedly into many identical RNA molecules, and each mRNA can be translated repeatedly into many identical RNA molecules, and each mRNA can be translated repeatedly to yield many identical polypeptide molecules. Remember that the final products of some genes are not polypeptides but RNA molecules that don't get translated, including tRNA and rRNA. In general, the steps of transcription and translation are similar in bacterial, archaeal, and eukaryotic cells. The major differences is the occurrence of RNA processing in the eukaryotic nucleus. Other significant differences are found in the initiation stages of both transcription and translation and in the termination of transcription. In the first stage, transcription, RNA is transcribed from a DNA template, and RNA processing in eukaryotes, the RNA transcription pre-mRNA is spliced and modified to produce mRNA, which moves from the nucleus to the cytoplasm. The mRNA leaves the nucleus and attached to ribosomes. Ribosome amino acid activation. Each amino acid attached proper tRNA to its proper tRNA with the help of a specific enzyme and ATP. In translation, a succession of tRNA add their amino acid to a polypeptide chain as the mRNA is moved through the ribosome, one codon at a time. When completed, the polypeptide is released from the ribosome. The changes in genetic information of a cell called mutations, which are the ultimate source of new genes. They are responsible for the huge diversity of genes found among organisms. One of small-scale mutations of one or few nucleotide pairs, including point mutations, changes in a single nucleotide pair of a gene. If a point mutation occurs in a gamete or in a cell that gives rise to gametes, it may be transmitted to offspring and to future generations. If the mutation has an adverse effect on the phenotype of a person, the mutant condition is referred to as genetic disorder or hereditary disease. For example, we can trace the genetic basis of sickle cell diseases to the mutation of a single nucleotide pair in the gene that encodes the beta globulin polypeptide of hemoglobin. The change of a single nucleotide in the DNA's template strand leads to an altered mRNA and the production of an abnormal protein. In individuals who are homozygous for the mutant allele, the sickling of red blood cells caused by the altered hemoglobin produces the multiple symptoms associated with sickle cell disease. Small-scale mutations within a gene can be divided into two general categories. single nucleotide pair substitutions, and nucleotide pair insertion or deletion. A nucleotide pair substitution is the replacement of one nucleotide and its partner with another pair of nucleotides. Base substitutions may be a change in nucleotide pair may transform one codon into another that is translated into the same amino acid, such as change is an example of silent mutation, which has no observable effect on the phenotype. Substitutions that change one amino acid to another one are called missense mutation. Such a mutation may have little effect on the protein. The new amino acid may have properties similar to those of the amino acid it replaces, or it may be in a region of the protein where the exact sequence of amino acid is not essential to the protein's function. Substitution mutation are usually a missense mutation, that is, the altered codon still codes for an amino acid and thus makes sense, although not necessarily the right sense. But a point mutation can also change a codon for an amino acid into a stop codon. This is called nonsense mutation, and it causes translation to be terminated prematurely. The resulting polypeptide will be shorter than the polypeptide encoded by the normal gene. Uh, most nonsense mutations lead to non-functional protein. Insertion and deletion are additions or losses of nucleotide pairs in a gene. These mutations have disastrous effect on the resulting protein more often than substitution do. Insertion or deletion of nucleotides may alter the reading frame of uh, the generic message, the triplet grouping of nucleotide on the mRNA that is read during translation. Such a mutation, called frameshift mutation, occurs whenever the number of nucleotides inserted or deleted is not a multiple of 3. Mutations can arise in a number of ways. Errors during DNA replication or recombination can lead to nucleotide pair substitution, insertions, or deletion, as well as to mutations affecting longer stretches of DNA. If an incorrect nucleotide is added to a growing chain during replication, for example, the base on that nucleotide will then be mismatched with a nucleotide base on the other strand. In many cases, the error will be corrected by DNA proofreading and repair system. Uh, Otherwise, the incorrect base will be used as a template in the next round of replication, resulting in a mutation. Such mutations are called spontaneous mutations. A number of physical and chemical agents called mutagens interact with DNA in ways that cause mutations. In the 1920s, Hermann Muller discovered that X-rays cause genetic changes in fruit flies, and he used X-ray to make Drosophila mutants for his genetic studies. Mutagenic radiation, a physical mutagen, includes ultraviolet light, which can cause disruptive thymine dimer in DNA. Chemical mutagens fall into several categories. Nucleotide analogues are chemicals similar to normal DNA nucleotides, but that pair incorrectly during DNA replication. Other chemical mutagens interfere with correct DNA replication by inserting themselves into the DNA and distorting the double helix. Researchers have developed a variety of methods to test the mutagenic activity of chemicals. A major applications of these tests is the preliminary screening of chemicals to identify those that may cause cancer. This approach makes sense because most carcinogens, cancer-causing chemicals, are mutagenic, and conversely, most mutagens are carcinogenic.